Good afternoon, learners. Welcome you all to the Gyan Darshan session. Now, if you remember, in my last session, I started discussion on the M.Sc. Zoology program laboratory course 1, which is the practical course in your M.Sc. Zoology program. And in my last discussion, I took up 3 experiments, one on plasmid isolation, one on agarose gel electrophoresis, and also the quantification of the nucleic acids. So these 3 experiments were dealt in my last discussion. Today, we will proceed with further 3 experiments of the same course, that is laboratory course 1. And today we will be discussing on the restriction enzyme digestion, ligation and bacterial transformation, preparation of competent cells and bacterial transformation. So these experiments are again part of your molecular biology, the basic discipline of molecular biology. And we will take up one experiment one after the other. So we will start with the restriction enzyme digestion. So in my last discussion, we learned how to isolate plasmid DNA, how to visualize the plasmid DNA in agarose gel, and how to quantify the nucleic acid or the isolated DNA, particularly regarding the quantification of the nucleic acid, quantification of DNA, and measuring the purity of the DNA, isolated DNA. So once these are all properly checked, the next experiment that we will learn today is how to digest the DNA. So all of you must have heard about the restriction enzymes. So before we go into the experiment, let me first briefly discuss what is meant by a restriction enzyme and why do we need restriction enzyme in molecular biology. Now, restriction enzyme digestion, restriction enzymes are basically the enzymes which cut the DNA at some specific sites. Now, they can be hexacutters, they can be tetracutters. Hexacutters means which can recognize a hexanucleotide sequence and cut at one specific nucleotide. So they make site-specific cuts in the DNA. So it's a process, restriction digestion is the process of cutting DNA molecules into smaller fragments with special enzymes called restriction endonucleases. These are actually bacterial enzymes which cut DNA at specific sites to produce defined and reproducible fragments. And most of the restriction enzymes that we use in molecular biology belong to the type 2. Actually, there are different groups of restriction enzymes which probably you will be studying in your genetics and animal biotechnology course of your M.Sc. Zoology program. And there you will be studying the details of all these restriction enzymes. But since we are discussing the practical part here, so we will not be elaborating too much about the theoretical part. Rather, we will learn how to cut the DNA, how to do the practical part. So type 2 restriction enzymes are mostly commonly used in molecular biology, and they bind DNA at specific recognition site consisting of short palindromic sequences. Now, what is meant by palindromic sequence? That means the sequence, if you read the sequence from 5' to 3' direction, it will read the same if you read it from 3' to 5'. So that is known as the palindrome. So if you read the sequence from 5' to 3' and if you read the sequence from 3' to 5', it will read the same. Now, the digested DNA, once you cut the DNA with restriction endonucleases, then you need to observe the DNA, whether you have properly cut the DNA or not, and for that you have the agarose gel electrophoresis. So what we will learn in this experiment, we will learn how to cut the DNA, number 1, using a particular enzyme, and secondly, we will learn how to visualize the DNA, whether the DNA has been properly cut through agarose gel electrophoresis. So what are the things which are very important in this experiment? Number one is how much amount of DNA you should take. Okay. Number two is how much enzyme you should take. Number three is how will you set up a reaction? I mean, you need a reaction mixture, and in that reaction mixture, you need to add the components, and then you need to incubate the mixture, and then observe whether the DNA has been properly digested. So these are the three main components of this experiment. Of course, you know that the plasmid, it has a multiple cloning site where there are several restriction enzyme sites. So you can choose any of these enzymes to cut the plasmid DNA. Again, when you cut the plasmid DNA, you can use one enzyme or you can use two enzymes also. Like if you want to cut the plasmid DNA with two enzymes, let's say BAMH1, EcoR1. Or if you want to digest the plasmid with one single enzyme, maybe EcoR1 or Hindi3 or BAMH1, whatever. So either you can go for a single cut or you can go for a double cut. And the effect will be like the plasmid DNA will be linearized. When you cut it, it will be linearized. That means if you run the agarose gel, it will show one single band. Because if you remember, in my last discussion, I said that when a plasmid DNA is run in an agarose gel, normally there are three bands. That is supercoiled, linear, next circular. Here, you will observe only one band. That is, if you cut the DNA with either one enzyme or two enzymes, you will observe one single band, that is linear band. So that is how we will be visualizing it in an agarose gel. Now, why do we learn actually? What is the need of learning this experiment, or what is the need of knowing the experimental protocol? Because you know that in molecular biology, one of the very important component is cloning, where we clone a foreign DNA into a vector molecule or a plasmid molecule. So in order to clone, you need to cut. You need to cut the plasmid DNA as well as you need to cut the foreign gene or the foreign DNA with the same enzyme so that it generates a sticky end and we can clone it easily into the vector. So you have to cut the vector molecule as well as you have to cut the foreign gene or the insert. We call it insert. We have to cut it with the same enzyme, and thereafter you can go for the cloning, which is the next experiment that we will discuss today, that is ligation. So if that background is clear in our mind, and as I said that we are not discussing much on type 1 and type 3 because these are not that much used. We normally use type 2 enzymes, and most of the type 2 restriction enzymes, they either make staggered cuts or they make blunt cuts. Like there are some enzymes which generate blunt ends and there are some enzymes which generate sticky ends, or they make staggered cuts. So that is actually the theoretical background. Now we come to the experimental part, that is how will you actually set up the reaction in a laboratory. If you undergo this experiment, how will you set up the reaction? Now, procedure. So what you have to do, you have to take an Eppendorf tube and you have to take all the components. What are the components that are necessary for setting up a reaction mixture? You need a restriction enzyme buffer, because any enzymatic reaction will need a buffer. So you need a buffer. You need the enzyme itself. You need water, okay, and of course you need the DNA, that is the plasmid DNA or the insert DNA, whatever be the case. So you have to take the requisite amount of plasmid DNA in an Eppendorf tube. Now if the question that comes to mind is how much DNA should we take? You have already learned how to quantify DNA, that is spectrophotometrically. So normally 500 nanogram to 1 microgram of DNA may be taken. So this is a range, and for different purposes, we normally, for different plasmids, we have to use different concentration of the plasmid DNA. Then what you have to do is you have to add the reaction buffer. Normally the buffers are available in 10x concentration, and you have to use it at 1x concentration. So if you are having a reaction mixture of total volume 50 microliter, it means that you have to take the buffer 5 microliter because it is 10x and you have to use 1x. So if you set up a 50 microliter reaction, you have to take the buffer 5 microliter. You have to take the restriction enzyme. The restriction enzymes, the activity of a restriction enzyme is expressed as, like I have shown in bracket, 1 unit per microliter or 5 unit per microliter. So that is actually the specific activity of a restriction enzyme. And you have to add the restriction enzyme. You have to add the buffer. The rest of the volume should be, you have to use the sterile water or the autoclaved water, and the plasmid DNA like 500 nanogram to 1 microgram. And then the reaction, you have actually made up the reaction mixture. And now you have to incubate the reaction mixture at 37 degrees centigrade for 3 hours. Sometimes in certain cases, like if you are digesting a genomic DNA and not a plasmid DNA, you might have to increase the incubation period. Sometimes even overnight, you might have to incubate the reaction mixture. But for plasmid DNA, normally the incubation time is 3 hours, maximum 3.5 hours. So the reaction conditions vary for different enzymes. So this is what you can see, that how you are setting the reaction mix, as I told, that is enzyme buffer. Like here I have shown 25 microliter reaction mixture. So I have taken buffer 2.5 microliter, restriction enzyme 1 microliter, DNA 500 nanogram to 1 microgram, sterile water as required. And you make up the volume to 25 microliter. You can even make up the volume to 50 microliter. In that case, you have to use 5 microliter buffer, as I said. Now, what is meant by this 1 unit of restriction enzyme? Because I have written like 1 unit per microliter or something like that. I have written, if you see, 1 unit per microliter. What is meant by unit? 1 unit of enzyme means it can digest 1 microgram of DNA in 1 hour. So supercoiled plasmid DNA generally require more than 1 unit for complete digestion. So it might vary, like you might need 1 microliter, or you might need 1 unit, or sometimes even you might need 2 units. So it will actually vary with the plasmid. It will also vary with the purpose of your experiment, purpose of your study. It depends on the copy number also sometimes. That is, if it is a low copy plasmid, then it's a different. If it is a high copy, then it might differ. So there are several conditions. So that is how my main intention is to let you know how to set up a reaction, what are the components that are added, and what is the temperature that you need to maintain, and how many hours you need to maintain the reaction. So that is how you have actually set up a reaction. Now the next part is, after that you have set up the reaction and you have incubated it for 3 hours, next task is you have to check whether the DNA has actually been digested or not. For that, you have to again go for that experiment which we discussed in the last session, that is agarose gel electrophoresis. So for those who have not followed that session, for them I will just recap it in brief once more, that what is meant by agarose gel electrophoresis. Now before I go into that, you can just take a look at the observation. That is, this is actually how it will look like. You can see a series of bands. So which means that the DNA has been digested. Okay, sometimes it might happen that a fragment is released after digestion. There is a fragment, maybe that one piece of fragment is released because of digesting the plasmid DNA. So in that case, you will notice two bands. One is the main band and another is the released band, which will be of lower molecular weight and which will come at the lower portion of the agarose gel. Okay, so it will vary, but if it is just a plasmid DNA and you cannot detect the fragments as such, then you will get one single band. So, and how will you check whether your desired fragment has been released? Like if you want to release any fragment by cutting a plasmid DNA or any DNA, if you want to release a fragment and if you want to check whether the appropriate size fragment has been released, then you have to use a marker. As you can see in lane number 1 and lane number 6, these are actually the molecular weight markers. That means usage of this marker actually implies that there are certain fragments which are already there of known molecular weight, like 4840, 3590, as you can see on the left side, 2810. These are the fragments which are already known. And so you can compare your fragment, okay, with respect to these fragments. And so you will be able to understand what is the size of your fragment that is released from your plasmid DNA. I mean the DNA that you are using in your experiment. So you can compare against this standard and you can at once tell that what is the fragment size of the plasmid DNA that you are using. Okay, and you have digested it and what is the released fragment size. So that is the utility of using these molecular weight markers. The rest of the things could be well understood, that they are digested and therefore certain fragments may be released, or sometimes they are just, the plasmid DNA is just linearized, showing one single band. Now this you are monitoring through an agarose gel, and let me just recap once more what is agarose gel electrophoresis, what is the principle. Like agarose gel electrophoresis, this is a method for separating the nucleic acids, DNA, even RNA. And you can visualize the DNA and RNA through agarose gel. And here the gel is run in a horizontal fashion and not vertical. So there are different factors which determine the mobility of DNA in an agarose gel. So number one is the agarose concentration. That is, if you use a high agarose concentration, then the migration will be slower. If you use a lower agarose concentration, migration will be faster. Now what is agarose? Agarose is a linear polymer composed of alternating residues of D and L galactose joined by alpha-1,3 and beta-1,4 glycosidic linkage. And aggregation of helical fibers formed from agarose chain lead to supercoiled structure with 20 to 30 nanometer radius. So migration depends on number one, agarose concentration, agarose type, DNA conformation. Supercoiled DNA will be the fastest running, linear form in between, and the Nick circular will be the band which is the slowest. So first, the fastest will be supercoiled, then would be linear, and then Nick circular. Size of the DNA. If the DNA molecule is larger, then the migration will be slower, and vice versa. If the DNA is smaller, base pair is less, it will migrate faster. So the relation is, it is inversely proportional to the logarithmic 10 of the number of base pairs. Electrophoresis buffer. Because you know that water is a poor conductor of electricity. Electrical conductivity in absence of ions is minimal. So you have to use a buffer which is of particular ionic strength. But remember that if you have a very high ionic strength, then also a large amount of heat will be generated. Therefore, normally electrophoresis buffer that we use is tris acetate EDTA. Voltage. If you apply a very high voltage, what will happen? The DNA will migrate faster, no doubt, but again a lot of heat will be generated. So again, if the voltage is very low, then also migration will be very slow. Therefore, you have to optimize the applied voltage. Normally for DNA, it's 70 to 80 volts that we use for running a particular DNA molecule. Ethidium bromide in the gel. If you add ethidium bromide in the gel or in the buffer, then there is a slight retardation. Migration rate, it is retarded by a factor of around 15%, 1.5. But then you have to use ethidium bromide because ethidium bromide is an intercalating dye and DNA can be visualized only after you stain it with ethidium bromide. So you have to use ethidium bromide, but better not to use it directly in the gel or directly in the buffer. You can use it as a staining agent. After the gel run is over, you can stain the gel in ethidium bromide for a short period of time and then visualize it under UV transilluminator because it's a fluorescent dye. As I said, that electrophoresis buffer, that is tris acetate EDTA, pH 8 is normally used. Another thing which you should remember is when you are loading the DNA in an agarose gel, you have gel loading buffer. And this gel loading buffer normally consists of dye, that is bromo evenly into the well. It adds color to the sample so that you can track the migration of DNA. How much the DNA has entered in the agarose gel, that you can track. And number 3, they contain dye that move towards the anode in an electric field. So what are the dyes that you use in this gel loading buffer? That is bromophenol blue and xylene cyanol. Normally, bromophenol blue, the migration rate of bromophenol blue is 2.2 fold faster than xylene cyanol and corresponds to 300 base pair DNA. That means bromophenol blue will migrate faster as compared to xylene cyanol. And normally bromophenol blue, wherever you will find the bromophenol blue in the agarose gel, you can understand that it will correspond to around 300 base pair DNA. And wherever you will find xylene cyanol FF in the agarose gel, we will understand that it corresponds to 4 kilobase pair DNA. So in that way, if you know what is the molecular weight of your DNA sample, then you can at once decide how much running time should you apply. Because you are sure that wherever you will find bromophenol blue, that is 300 base pair. So if your DNA is around 500 base pair, then make sure that the bromophenol blue does not come out of the gel. Okay? And even if the bromophenol blue is in the mid of the gel, you will be able to track the 500 base pair DNA. So in that way, you can decide. And finally, when the gel run is over, I already told that you need to stain the gel because you need to stain the DNA, and for that you have to use ethidium bromide, which is a phenanthridine monomer fluorescent dye which intercalates between the stacked bases. And you have to observe the DNA under UV transilluminator. That is, DNA absorbs UV radiation at 254 nanometer. To be precise, we say 260 nanometer. To be more accurate, 260 nanometer and precisely 254 nanometer. So in case of plasmid DNA, nucleic acids are electrophoretically separated on an agarose gel, and then you stain the gel with ethidium bromide and visualize it under UV transilluminator. So that is how you undergo an agarose gel electrophoresis, and these are actually the steps. I think I showed the same slide in my earlier session also. That is, you have a casting tray, that is glass plate. You pour the gel, agarose gel, allow it to solidify, and then put a comb to create the wells, and then you can load the DNA sample using the pipette man, which I already showed in the last session. And then you run the power supply. That is, the DNA will migrate from the negative to positive because DNA is negatively charged. And after the run is over, you have to stain the gel and visualize it under UV transilluminator. So the same principle also holds true for restriction enzyme digestion. So you cut the DNA, as I said, and then you load the DNA in the agarose gel and you visualize whether the digestion is successful or not. Either you may get a single band, that is linear band, or you may get 2 bands or 3 bands depending on whether a fragment of a particular size has been released from the plasmid or not. So that is the total experiment for the restriction enzyme digestion followed by agarose gel electrophoresis. So we now move to the next experiment, that is ligation of insert DNA in the vector, the second experiment for today's discussion. What is meant by ligation? Ligation means joining. Now once that you have digested the plasmid DNA and once you have digested the foreign DNA fragment also, you need to join them. And for that joining, you need one enzyme which is called DNA ligase. Now again, this is an enzyme. So ligation into the multiple cloning site of any vector is one of the most important steps in the cloning process. The joining of DNA fragments through recombinant DNA technology is essential for many processes, including sequencing reaction, protein expression, mutagenesis, gene analysis, etc. So how will you undergo this experiment? So you have digested the DNA. You now have learned how to digest the DNA. Now you need to join. So again you will need a reaction mixture. Now in this reaction mixture, what you are going to add? Number one, you need the DNA again. That is the plasmid DNA, and also you need to add the insert DNA. So the question is, how much plasmid DNA should I add and how much insert DNA should I add? That is, the foreign gene you need to add and the vector molecule you need to add. So how much? So normally for cloning or for ligation, we normally use 100 to 500 nanogram of the digested plasmid DNA, restriction enzyme digested plasmid DNA. However, it might vary, because if it is a low copy vector, it might be even you might have to take much higher amount of the plasmid DNA. It could be 1 microgram or 2 microgram also. But if it is a high copy plasmid, then even you can use 100 nanogram. 100 to 500 nanogram can be quite sufficient. So that is about the vector molecule. What would be the insert molecule? Now here there is one rule. That is, the insert molecule should be 3:1 molar excess over the vector molecule. So you have to calculate the molar excess. Now what is meant by this molar excess? You have to understand that very clearly. Like I gave you an example here. If you are ligating a 1 kilobase pair fragment into a 3 kilobase pair plasmid, okay, so the plasmid is 3 times larger than the foreign DNA. But what we need is, we need the foreign DNA. How much excess? 3-fold molar excess. Because we said 3:1. So the 100 nanogram of the plasmid can ideally accept 33 nanogram of the insert. But we have to use 3-fold excess of the insert. That means 33 into 3, that is 99 nanogram, which is almost equivalent to 100 nanogram. So in this case, if you are ligating a 1 kilobase pair fragment into a 3 kilobase pair plasmid, and if your plasmid DNA is 100 nanogram, then your insert DNA in this case will also be 100 nanogram, because 99 nanogram and 100 nanogram is close. Having a slight excess of insert makes it more likely that it will bump into the plasmid and the ligase, and it is more likely to ligate to other copies of itself or give you plasmids with multiple copies of the insert back to back. So there are certain disadvantages. If you don't calculate it properly, then there might be chances that your ligation experiment will fail. So normally it is recommended that you use 2:1 molar excess, maximum 4:1 molar excess. You can never go beyond that. Ideally 3:1 molar excess, that is insert is to vector, is recommended. So you need to know the molecular weight of the plasmid, the molecular weight of the insert, and then calculate the molar excess properly, calculate the how much amount you have to add, calculate it properly, and then go for the reaction. So that is actually the main principle here to set up a ligation reaction. Otherwise, it is not at all very difficult, because again you need to set a reaction mixture. You can set it at 20 microliter. So again you have a 10x buffer. So if you are setting up a 20 microliter reaction, you have to take 2 microliter of the buffer, because you are using the buffer at 1x concentration and the stock concentration is 10x. You have to add the ligase enzyme. Again the ligase enzyme will come as like 5 units per microliter or 1 unit per microliter, whatever. So you can add 1 microliter of the T4 DNA ligase enzyme. Buffer I already said. Vector DNA you have to add, insert DNA you have to add, and the rest will be sterile water. So you have to add the reagents in the following order: water, buffer, insert, vector, ligase. So here I have shown that you are setting up the reaction at 20 microliter. Final volume is 20 microliter, and then you have to incubate the reaction at 20 degrees centigrade for 4 hours or 12 to 16 hours at 16 degrees centigrade, whichever be comfortable for you. So that will ensure that the foreign DNA has been joined or ligated with the plasmid DNA, that is vector DNA. Now in this context, I would like to mention another case, that is sometimes during the ligation or prior to ligation, you might have to treat the plasmid DNA with another enzyme which is known as CIP, that is calf intestinal phosphatase. Now what is the function of a phosphatase enzyme? Phosphatase means which will remove the phosphate group. Where is the phosphate group located in the DNA? That is at the 5' end. So this enzyme will actually remove the phosphate group from the 5' end of the DNA. Why do you need this removal? Why do you need to treat the plasmid DNA with CIP before going for the ligation? Because sometimes it might happen, especially when you are cutting the plasmid DNA with one single restriction enzyme, there is a chance of self-ligation. That is, the plasmid DNA, because the sticky ends are generated at the two ends, the plasmid DNA will tend to recircularize. Okay, it will lead to self-ligation. If you add the ligase enzyme, then it will self-ligate and it will not incorporate the foreign DNA fragment. But if you remove the phosphate group, then the chances of self-ligation is reduced. There is lesser chance of self-ligation. I won't say that it is totally not there. Maybe some DNA might still undergo circularization, self-ligation, but the chances are less. And so there is more probability that your foreign DNA will be incorporated into the plasmid DNA. So this is also particularly when you are cutting it with one single enzyme, make sure that you are treating the DNA first with CIP, calf intestinal phosphatase, and then inactivate the enzyme, heat inactivate the enzyme, and then go for the ligation reaction. Otherwise, you will come up with all self-ligated vectors and not those vectors which have incorporated the foreign gene. So that is all about the ligation reaction. Now the question is, how will you check whether the ligation has been properly, you have properly undergone the ligation reaction or not? Now just take a look at the steps of ligation process which I have shown here. That is, you are cutting the DNA, you are mixing it with the foreign gene, and then you are ligating it. See, the red part is the foreign gene, red fragment is the foreign gene. So you are cutting, the purple one is released from the plasmid, and in that place you are incorporating your foreign gene, that is the red one. And if you are doing the CIP treatment, then you have to treat it with phosphatase and followed by ligase, so that there is no chance of self-ligation. When you add ligase, there is no chance of self-ligation. Rather, when you add ligase and the DNA insert, it is the DNA insert which is incorporated and not the recircularized self-ligated plasmid. So that's what I was discussing. But the last part of the experiment, that is preparation of competent cells and bacterial transformation. Now, why do we need this experiment? Whether your DNA has properly undergone, your ligation reaction is properly successful or not, that will be dictated by, or that can be monitored only through transformation. Moreover, what is the purpose of knowing transformation? Like supposing you need to incorporate a plasmid DNA in a bacterial cell. So in that case, what you have to do is you have to undergo transformation. What is meant by transformation? It is the uptake of foreign DNA by a bacterial cell. Now, the bacterial cells, normally they don't take up foreign DNA so easily. Rather, the bacterial cells should be made competent. What is meant by competent? Competent means they should be made, they should be treated in such a way that they are capable of taking up the foreign DNA. Normally they don't take up foreign DNA so easily. But you have to do the experiment. Like you have undergone ligation and you need to check whether the ligation is successful or not. What you have to do is you have to incorporate the ligated DNA into a bacterial cell, will be capable of growing in an antibiotic medium, selection medium. The bacterial cells which have not taken up the plasmid DNA will fail to grow in the antibiotic medium. And therefore, you can screen easily that your ligation reaction is successful. So what is meant by competent? Competent cells means you have to create some alteration in the cell wall of the bacteria. Okay, and so the foreign DNA can easily enter or migrate through the cell wall and enter within the cell. Now, the normal procedure which we use in the laboratory for making a cell competent is through calcium chloride treatment, followed by a heat shock treatment. So this is the basic principle of undergoing bacterial transformation. That is, treat the bacterial cell first with ice cold calcium chloride, okay, and then decant the calcium chloride off, add the plasmid DNA, keep it on ice, and then subject it to a brief heat shock. That is, heat shock is normally applied at 42 degrees centigrade. Okay, and then suddenly you bring out the tube and place it again on ice. So this sudden heat shock followed by a cold shock, that is heat shock followed by cold treatment, it creates some sort of jerk and it perturbs the cell wall and also the membrane, so that the DNA is capable of entering into the cell, because there are certain minute pores which are created. Of course, there are several hypotheses which are associated with this treatment process, that what is actually happening. There are a lot of, explanations, and there is no single explanation that can explain the bacterial uptake, the foreign DNA uptake by a bacterial cell. So bacterial cells, they show natural competence, but those lacking natural competence can also be made competent artificially. Not, as I said, bacterial cells, they don't take up DNA so easily. They have to be treated. They have to be made competent. And then only they are capable of taking up the foreign DNA. DNA is a highly hydrophilic molecule and normally cannot pass through the bacterial cell membrane. In order to make bacteria capable of internalizing the genetic material, they must be made competent. Treatment with calcium ions is the standard method for the preparation of competent cell. Suspension of bacterial cells in a high concentration of calcium solution create minute pores through which the extra chromosomal DNA will be forced to enter. Thereafter, it is followed by a brief heat shock treatment. I already mentioned. So first calcium chloride treatment followed by heat shock treatment. What happens here is the DNA molecule binds to the lipopolysaccharide receptor molecules on the competent cell surface. Because if you are doing the experiment with E. coli, which is the common model used, then E. coli is a gram-negative bacteria and it has the lipopolysaccharide molecule. So DNA molecule binds to such lipopolysaccharide and then addition of calcium chloride to the cell suspension allows binding of the DNA to the lipopolysaccharide. And this heat shock, brief heat shock, it actually creates depolarization of the cell membrane of calcium treated cells. So calcium treatment, creation of pores, followed by heat shock, which creates depolarization of the membrane, and altogether they facilitate the entry of the DNA molecule within the cell. So if that principle is clear in your mind, then we can go for the protocol or the procedure part. That is, how will you undergo the experiment now in the laboratory? So procedure is, what you need is, you need to first grow the E. coli cells. Grow the E. coli cells overnight and derive a liquid culture. Overnight liquid culture. Maybe in a 5 ml medium. The media which are normally used is the Luria-Bartani medium. Okay. And this Luria-Bartani actually consists of peptone, yeast extract and sodium chloride. Back to peptone, back to yeast extract and sodium chloride. So that is actually the composition of the Luria-Bartani broth or growth medium. So you have to grow the bacterial cells in Luria-Bartani medium. You get an overnight culture. Next day, that is day number 2, what you have to do is add 1 ml of this overnight culture to 50 ml of Luria-Bartani liquid medium. Again you grow the culture for 2 to 3 hours and then measure the optical density of the culture in a spectrophotometer at 600 nanometer. And if you see that the optical density, it reaches 0.4, then you can understand that it is suitable for bacterial transformation, because it has almost 10 to the power 8 cells per milliliter. So if you have 10 to the power 8 cells per milliliter, it will correspond to around OD 600 0.4. What you will do next? You will transfer the culture in a centrifuge tube, cool the culture on ice for some time, and then centrifuge it at 5000 revolutions per minute at 4 degrees centigrade. Be very careful that all the steps should be performed in ice cold condition. You need ice bucket where you need ice, and you need a cold centrifuge machine where you can set the temperature to 4 degrees centigrade, because you cannot undergo the reaction at room temperature or even at higher temperature, because in that case the bacterial cell, because of there will be a heat shock, and the bacterial cells will not be able to take up the DNA, because you need the cold treatment. Specifically, you need the cold treatment, calcium chloride cold treatment, for creating the minute pores. And you cannot subject the bacterial cell to prior osmotic shock by exposing it to room temperature or high temperature. So After the bacterial cells are being pelleted down, you add 5 ml of calcium chloride, ice cold calcium chloride, and centrifuge it at 5000 revolutions per minute at 4 degrees centigrade for 5 minutes. Okay, so you decant the calcium chloride and then resuspend the pellet in 1 ml of ice cold calcium chloride, and then dispense the bacterial suspension into Eppendorf tubes. Let's say 200 microliter aliquots in separate Eppendorf tubes. So day number 2, work for day number 2 is done. Now day number 3, third day. What you will do here, that you need to add the DNA now. You already have the plasmid DNA, or you already have the ligated DNA, that is the ligation mixture you already have. Or if you are not transforming ligated DNA, you already have the plasmid DNA. So you add the DNA to the bacterial suspension. Let's say 200 microliter aliquot you add. Keep it on ice for some time. And then subject it to heat shock 42 degrees centigrade for 90 seconds. Exactly 90 seconds. Don't exceed the time. So 42 degrees centigrade and 90 seconds, these two are very important and you have to properly maintain it. So there is a heat shock. Once the heat shock is over, immediately place the tube on ice. So heat shock followed by a sudden cold treatment. So you can keep it on ice for 10 minutes. Okay, so that is done. Now what you do is, you have to add Luria-Bertani liquid medium, which you have already prepared at the start of the experiment. You add 500 microliter of Luria-Bertani liquid medium to that bacterial suspension, where you have added the DNA and subjected it to heat shock. And then you leave the mixture in an incubator shaker at 37 degrees centigrade for 45 to 60 minutes, around 1 hour to 2, it can be also 2 hours, 1 hour to 2 hours. Next what you do is, you have to spread the 100 microliter of the sample into agar plate, Luria-Bertani agar plate, that is solid medium. And that plate should contain the desirable antibiotic. Which antibiotic? The antibiotic resistant gene that is present in the plasmid. If your plasmid has ampicillin resistant gene, then the agar plate should have ampicillin as the antibiotic. So you are spreading the entire transformed sample or the bacterial suspension into the LB, that is Luria-Bertani agar plate containing the desirable antibiotic, and leave the plate at 37 degrees centigrade overnight for 18 to 20 hours. Next phase, in the next phase, what you will be able to see? You will be able to see bacterial colonies, like as I am showing it here. What does the appearance of these bacterial colonies suggest? Suggest that ligation followed by transformation is successful, or transformation is successful, because the bacterial colonies, they are capable of growing in the antibiotic medium, which means all these bacteria, they have the desirable plasmid which you wanted to transform. And the transformation efficiency will be given by this formula, that is number of colony forming units, abbreviated as CFU, divided by amount in microgram of plasmid DNA. So you can calculate the colony forming units and you can calculate the transformation efficiency. That is how efficient your transformation process is, or you have undergone the transformation experiment. So that is actually the principle. And finally, when you see your plate, that is LB agar plate, Luria-Bertani agar plate, you will be able to find out colonies like this. But if your plate does not contain any colony, that means somewhere the transformation is not properly done. Sometimes it may happen that the transformation efficiency is very low. You might not see so many colonies. You might see maybe some 50 colonies or 25 colonies. It is possible, especially when you are undergoing ligation, checking the ligation reaction, and you have transformed a ligation mixture. It might happen that the number of colonies might be less. So that is how you actually undergo this transformation experiment. So these were the three main experiments that we discussed today. So if I recap once more that what we actually learned today. So actually if you take the two sessions, last session and today's session together, it actually makes a complete story, because first you learned how to isolate a plasmid DNA. Next you learned how to check the plasmid DNA through an agarose gel, that is agarose gel electrophoresis. Then you learned that how will you quantify the nucleic acid, how will you quantify the DNA, that is what is the concentration of the DNA, and whether the DNA is pure DNA, because I told you that we measured the OD260 by OD280 ratio, and that is how we check the purity of the DNA. Once these are done, then you may go a step forward where you may digest the DNA as we discussed today, that is digest it with a restriction enzyme. Why you need to digest? Because you need to clone some foreign gene. If you need to clone a foreign gene, you need to cut that gene also, and you need to cut this vector also. And finally, for the joining, what you need is the ligation, where you are using a ligase enzyme. But again, you have to check whether ligation is successful. That you can do via bacterial transformation. So you just, what you do is you just try to mobilize the ligated mixture into a bacterial cell by treating the bacterial cell with calcium chloride followed by heat shock, which creates a favorable condition for the DNA entry. And then what you do is you need to plate the bacterial cells which have already uptaken the DNA in an antibiotic selection medium and grow the plates at 37 degrees centigrade. The next phase, in the next phase, you see bacterial colonies, which ends the total story of the gene cloning process, starting from isolation, cutting, to ligation and transformation. So with that, I will end for today's discussion, and in the coming sessions we will proceed with further experiments that you have in your course, especially components like cell culture, animal cell culture, and how will you check the necrotic, how will you check the apoptotic cells, how will you check DNA damage. So these are some of the experiments which will be followed up in the next sessions. So till then, please go through the course material, whatever we have discussed for these two sessions, and if you have any problem, you can always write to us, or you can contact us, email us, and we will be there to respond to all of your queries. Thank you all, and enjoy your learning.